All right, guys, this is part 2 of Roman numeral 4. So this is going to be about leukocytes, and then I think we'll finish up with platelets and clotting factors. So what you're looking at is the 5 different leukocytes that you guys will need to be able to recognize from their image. And it turns out it's not that hard. Lots of students get it right away, so don't be scared. Let's start with our neutrophil. So along with recognizing their picture, you also need to know what their sort of specialty is. Okay, so let's go through them. Their letter A and letter B, the leukocytes here are actually divided into 2 different categories. They are called granulocytes or agranulocytes. So it turns out that when you stain these white blood cells with the stains that they use, the cytoplasm either shows up grainy or does not. So early on, they divided these white blood cells into granulocytes, the ones that look grainy, and agranulocytes, the ones that don't. So out of this picture, it's actually quite difficult to tell, I think. I think they all look a little grainy, but the first 3, the neutrophil, the eosinophil, and the basophil, those are granular. Okay, so the cytoplasm is grainy. It's filled with vesicles that are also filled with chemicals that mediate allergic or different reactions. Allergy is one of them, but different immune reactions. The monocyte and the lymphocyte are considered agranulocytes. So their cytoplasm is a little bit less grainy in appearance, but it's a little bit of an artificial term because monocytes and lymphocytes are also carrying vesicles with chemicals inside. They just don't show up as dark, okay? So let's take a look at our granular leukocytes, letter A, our neutrophils being the first one. So our neutrophil is going to be called neutrophil because the cytoplasm is neutral in color. It doesn't really pick up a color. Now, I know you guys are saying it looks purple, but when you compare it to the other 2, the eosinophil and the basophil, the cytoplasms here definitely pick up a color, right? This is a pink cytoplasm. This is a dark blue or purple cytoplasm. And here they consider this neutral. Okay, so neutrophils are named after their neutral color of cytoplasm. Now, another word for neutrophil is, or another name, is polymorphonuclear cells. Polymorpho means many shape, right? Poly means many, morpho is shape, and the nuclear is the nucleus. So these neutrophils, their nucleus comes in really many different forms. There's not one distinct shape to their nucleus. So polymorphonuclear refers to that. They can have 2 lobes, 3 lobes, 4 lobes, 5 lobes. The lobes sometimes are connected by, little chromatin strings. And this one kind of just looks lumpy, okay? So what you want to know about these neutrophils is that they are mobile. They can exit the bloodstream and crawl around your connective tissues to find the infection. They are phagocytic, so they are going to phagocytose the foreign infectious particles, whether it's a bacteria, whether it's a fungus, whether it's maybe a dead cell. And so it's very phagocytic as well as mobile. Okay, and I have an asterisk next to the cells that are non-specific. What non-specific means is that these cells, and I'll just say right now that all 4 of these are non-specific. The neutrophil, eosinophil, basophil, monocytes are non-specific. The only specific one is the lymphocyte. What non-specific means is that these cells respond to anything foreign. Doesn't matter what it is. Staph bacteria, it can be E. coli bacteria, it could be a fungus, it could be whatever, right? It's going to respond to and gobble up, phagocytose anything foreign, non-specific. Let's just compare that to specific immunity, which is what the lymphocytes do. The T cells and B cells are both considered lymphocytes. They only respond to one particular antigen type. So remember I talked about antigens at the beginning of this lecture. It is a molecule that is recognized by the immune system. And out of all the B and T cells that you have in the body, every single one of them They can recognize a slightly different shaped antigen than each other. And so when you get infected with, say, a coronavirus, there's only going to be a small subset of B and T cells that recognize coronavirus antigen, but they do, which is the amazing thing, and then they will mount an immune response. So just the cells that recognize that specific antigen. So specific immunity is very specific, right? These cells only respond to one kind of antigen type. Let's just talk about this right now since we're on the subject. What is the advantage then of specific immunity, right? You might be saying to yourself, well, why limit these white blood cells as to like what they can recognize? Well, the nice thing is, since it is so specific, these cells also have the ability to remember. They have memory of the infectious agent. And so after you're infected with, let's say, coronavirus again, you retain memory to coronavirus. And so the second time you're infected with coronavirus, you have memory cells inside your body that respond much, much faster, much, much more effectively and stronger than the first time. And then it's such a huge immune response the second time that you don't get sick. So it wipes out the foreign entity immediately. So that's the huge advantage of specific immunity. What your T cells and B cells are able to do is this memory, okay? And this is what vaccines are predicated upon, right? So when you get a vaccine, you are stimulating the B and T cells of your body to make memory. And so in real life, right, when you are encountering the whooping cough bacteria or pertussis or whatever, and that's whooping cough. Came up with the same example twice. Or like the flu, right? You get a flu shot every year. Like you're giving your body that first encounter with the fake antigen, but it's going to stimulate that memory buildup. And so in real life, if you get infected with the real thing, your immune response is faster and much more intense, and so you won't get sick. Okay, so that's the benefit of specific immunity. Non-specific immunity is it's great also, right? You want your body to be able to get rid of anything that comes in. If you get a splinter under your finger, you're going to want, there's all kinds of microbes on that splinter. You want your immune system to fight all of them. So both of these kinds of arms of your immune system work together. Okay, so there's non-specific immunity as well as specific immunity. All right, so let's turn to eosinophils. So eosinophils pick up the stain eosin very, very strongly. Eosin is pink, so that's why the cytoplasm is pink. It's because of eosin. And the nucleus is going to be bilobed, so 2 lobes. If you look at your basophil, it's also a bilobed nucleus. So those are standard. The nucleus here is always going to be bilobed, even though sometimes it's not a pretty bilobe, it's always bilobed or 2 lobes. Same thing with basophil. Eosinophils are going to fight, their specialty is fighting parasites, and they're also involved in allergic reactions. So parasites are more like eukaryotic parasites. So so things like your standard parasite, like a tapeworm, these eosinophils will go way up, but also fungal infections. So eukaryotic fungus will also cause these guys to increase in number. So if you can look at your blood, if you have a really high count of eosinophils, then you might think, oh, this person might have a parasite or a fungal infection. Okay, let's look at basophils. So basophils are very dark. They are going to pick up that dark purple or blue stain. They also have a bilobed nucleus, but because the granules are usually so dark, it's very difficult sometimes to see the nucleus of a basophil. So they're just going to be really dark staining, really granular cells. And the hallmark of basophils is that these guys are really involved in allergy. And I have allergies right now, and these guys are driving me crazy because they're going to pump out histamine. These basophils pump out histamine, and histamine is the chemical that causes all those symptoms of allergy, because what histamine does is it vasodilates your blood vessels. So it causes the blood vessels in a particular area to get larger, that area then has all this liquid coming out, right? Because when you have a dilated blood vessel, more fluid comes in, more blood comes in, and then more fluid actually leaves, becomes leakier. So it leaves the blood vessel. So your area gets swollen. That causes swelling. Also causes redness and heat because of that blood. But histamine also causes itching and extra sensitivity to pain, and just all those lovely, itching and redness and pain and swelling, that's histamine. So that's why when you go and you get allergy medicine, they're antihistamines, right? Because you want to counteract the action of histamine. All right, let's talk about the agranular cells here. So monocytes. So these monocytes, their hallmark is a kidney bean shaped nucleus. Okay, so we don't really have two separate lobes connected by a little tiny thin chromatin. We have very broad kidney bean. Sometimes this nucleus will actually look a little bit more circular, but you're always going to look for this little dent or this notch in the nucleus. The cytoplasm is relatively clear. It's usually a very large cell with a lot of cytoplasm present. And these monocytes are in the bloodstream, but they will enter into your connective tissues. When they do that, they're called macrophages. We talked about macrophages before. So in the blood, they're called monocytes, but in the tissues, they're going to be called macrophages, and they're very phagocytic. These are the most phagocytic cell that you have. Macro means big, phage is to eat. They're big eaters. They're nonspecific, very phagocytic. Okay, and then our last agranulocyte is a lymphocyte. Lymphocytes have such a huge round nucleus that they only have a sliver of cytoplasm around that nucleus. So lymphocytes are pretty compact and small. You can see they're the smallest one out of the bunch. They're not much bigger than a red blood cell. Excuse me, but they're nice big round nucleus that takes up almost the whole cell, is going to give them away. Okay, so sometimes students get these two confused, the monocytes and the lymphocytes, but just look how much cytoplasm is here and how little cytoplasm is there, and then you always want to look for a notch as well. I will say sometimes a monocyte will get turned so that you don't actually see the notch. So imagine flipping this to its back, and if you're just looking at the back, but the other so then it might look round, but the giveaway is that you have a lot of cytoplasm, okay, when you compare the two. So let's take a look at this right here. So let's try and name each one of these, right? So we have this cell that has a grainy kind of cytoplasm that's sort of neutral in color. We have a very weird shaped nucleus. This is going to be our neutrophil. This is a kidney bean shape, very deep notch, a lot of cytoplasm. This is our monocyte. Excuse me. This is a very, very vibrant pink granules, bilobed, and this is our eosinophil. This guy is small, very round nucleus, little bit of cytoplasm seen. This is our lymphocyte. So lymphocytes are T or B cells. It's impossible to tell which one this is. It's a T or a B cell. In other words, just call it a lymphocyte. And then this one is so grainy, I can hardly make out the bilobed nucleus. This is our basophil. All right. So when we talk about number two, the lymphocytes, the B and T cells, the specific immunity, I mentioned that earlier, right? It has memory. And then I just want to go on and talk about B cells. B cells, these are the ones that create antibodies, okay? So out of your T and B cells, only B cells can create those antibodies. And then T cells have all different kinds of jobs, different than B cells. So T cells, what they do, their specialty is to recognize self versus non-self cells. So they can they can also recognize infected self cells. They can recognize your own cells that have been infected. So a virus, for example, coronavirus, right? It goes inside of your cells and it hides out there, and then it makes more viruses and it comes out. but your T cells can recognize a virally infected cell and destroy those virally infected cells. Pretty cool. T cells can recognize a cancerous cell. So they can recognize your cells as they go and become cancerous. There's different shapes and different proteins that they're expressing on the surface that are abnormal. T cells can recognize that and destroy your precancerous cells. So T cells are really good for infecting that. For destroying virally infected cells, cancer cells. They can also recognize foreign tissue. So when you have an organ transplant, let's say, it's the T cells that would recognize the foreign organ, not the B cells, and the T cells would reject them. So if you do have somebody who is getting an organ transplant, they have to take immunosuppressive drugs for the rest of their life because they need to suppress the T cells from rejecting the organ. Just one thing about the T cells, T helper cells are a very important kind of T cells. They coordinate every single part of your immune system. And the virus called HIV, human immunodeficiency virus, specifically kills these kinds of T helper cells. And so one of the hallmarks of HIV infection is your immune system is completely ruined. It's destroyed your immune system's response. And that's because the T helper cells are gone. And so that's sort of basically describes how important T helper cells are. If you wipe those guys out, you basically wipe out your entire immune system. So a person who has HIV is considered immunocompromised, which means that their immune system is weaker, right? All right, last subject here is our platelets. So this platelet image is, this is your bone marrow. This is a large cell called a megakaryocyte. And the megakaryocyte in your bone marrow will fracture, and those little fragments are called platelets. Okay, so I want to actually go and show you, sorry, I'm flipping through this for you, but show you the megakaryocyte in this step, right? If you didn't notice it before, I have it in red because I want you guys to know that this is the precursor of your platelets that are eventually secreted into the bloodstream. Okay, so let's go back to our lecture. All right, so platelets are going to help to create a clot, and what you're looking at is a blood clot. And this is where I show you the video in class, but I'm going to post the video to your module so that you guys can watch it. And I just want to talk through blood clotting. I'll just use this picture. So when you have a tear in a blood vessel, which is what a cut would be, right? When you start bleeding, that tear is going to signal the recruitment of your platelets and other things called clotting factors. Okay, so let's look at step number one, under hemostasis, which means to stop bleeding. If the blood vessel has a smooth muscle wall that's relatively robust, it will contract the smooth muscle and will, that's called a vascular spasm, to help to stop the bleeding. You know, the blood vessel itself is going to sort of pinch. That's not enough though, right? So then you have the first things that come in are called platelets. So the platelets will get activated. They're activated by the connective tissue fibers that are exposed when you have a broken blood vessel. So the platelets will only get activated if there's a broken blood vessel, which is pretty cool. So when they become activated, they become sticky and they start sticking to each other, and they're going to stick to not only the blood vessel, but to each other. And then other things are recruited. So this is going to start a cascade of events. And it's going to recruit things called clotting factors. So this is an activated platelet. Clotting factors, there's different kinds of clotting factors in your blood that are very, very important that we're not going to go over here. But there's another clotting factor that I talked about earlier, which is fibrin, right? So fibrinogen, which is floating around your bloodstream in the inactivated form, when there is a clot being formed, it gets activated to fibrin. And fibrin forms this mesh. So this is the stringy-like material that binds everything together. So look at the fibrin strands, right? This string that kind of nets over everything, that ties everything together. It reinforces the clot. And while the fibrin is weaving its clot, it's also trapping red blood cells and white blood cells within this big clot. So you can think of it, fibrin, as basically a fish net. So you threw a net over everything, catching different cells inside, and that eventually just becomes your clot. Okay, so the thrombus is the clinical term for a clot. Okay, a thrombus is going to have a mix of platelets or clotting factors and also trapped red blood cells. There is a disorder called an embolism. An embolism is where you create a clot, but then it breaks off the blood vessel wall, travels around your bloodstream, and it actually can get lodged and block the blood flow in a different blood vessel. So those are really dangerous. And then our last disorder is hemophilia. And hemophilia is a genetic disorder. It's relatively rare, but you are born with a deficiency in 1 or 2 clotting factors. So these are the clotting factors. It's not platelets, it's not fibrin, it's this other category called clotting factors. So if you have it, it just means that you have a problem clotting your broken blood vessel. So you may bleed for a longer time after an injury than you would if your blood clotted normally. Small cuts aren't much of a problem. The greater health concern is the deep bleeding inside your body, especially in your joints, knees, ankles, and elbows. And that internal bleeding might damage your organs and tissues and may be life-threatening. So think about if you had a small tear in your liver or in your kidney, but it just never stopped bleeding. That blood can accumulate and cause problems within that organ. So that's the issue here. So that's hemophilia, right? And at the moment, the therapy is to take clotting factors. So there are biological labs out there that create clotting factors. These are proteins that people have to take for life. Okay, so it's a lifelong treatment. All right, and that's it for blood.